transport chain, oxidative phosphorylation, and chemiosmosis is the heart of cellular respiration. It is during this process where the bulk of the ATP is produced through oxidative phosphorylation. This pathway is a series of cytochrome proteins embedded within the membrane of the mitochondrion. It is within the cristae of those mitochondria, which is why the cristae is in all these folds, increasing the surface area throughout. These proteins are embedded within this chain and actually do just as its name entails. It transports electrons from one protein to the next, all the way down to oxygen. These are electron carriers just like the NAD+ and FAD. Those coenzymes donate the hydrogens and electrons that they gather from our food sources, carry it all the way down the chain to oxygen. As those electrons are carried to oxygen, each coenzyme has the capability of producing ATP. One NADH can create 3 ATPs, whereas one FADH2 has only the capability of producing 2 ATPs. This is because FADH2 carries a slightly lower amount of energy. It's kind of like starting at the top of a hill and rolling down, and somebody starting in the middle of the hill at the same time. The person that starts at the top will reach the bottom of the hill with greater energy than the one in the middle because he was able to carry it further distance. That's exactly how NADH and FADH2 operate. This conversion of carrying electrons down the electron transport chain to oxygen is what fuels the chemiosmotic theory. If we examine the cristae real quick, we can see how it has all of these little folds within it. The NADHs and FADH2s are produced within the matrix of the mitochondrion. The cristae, however, has tremendous folds, and those folds actually have these cytochrome proteins embedded within them. If we look at this image here, what we see is the outer membrane of the mitochondrion across the bottom. Here we find the intermembrane space, and then the inner membrane of the mitochondrion with the folds of the cristae. These complexes here, 1, 3, and 4, or 2, 3, and 4, are cytochrome complexes. These constitute the electron transport chain. The NADHs are formulated or reduced in the matrix of the mitochondrion. It is in this matrix where we completely oxidize our glucose molecule. When those NADHs become oxidized again, they donate their hydrogens and electrons to the first cytochrome complex. The electrons are then carried from one protein to the next, all the way down to oxygen. The hydrogens, however, are actually pumped into the intermembrane space. This space here becomes highly concentrated in those hydrogen ions. And we all know what concentration ion gradients will do. They will go from a very high concentration to a very low concentration. But because hydrogen ions are charged, they must have a way to transport across the phospholipid bilayer. And that is where this complex comes in. This is what we call the ATP synthase enzyme. The hydrogen ions will flow through the ATP synthase enzyme back into the matrix of the mitochondrion. As they travel through this protein, this F1 complex here actually spins almost just like a cistern. As it is spinning, it rotates and rotates faster and faster, almost like a water wheel as well. What happens is this enzyme has binding sites for inorganic phosphates and ADPs. As the hydrogen ions flow through, the spinning of the cistern actually allows for that enzyme to produce the energy of ATP. If we look at these cytochrome complexes, you can see here's complex 1, here's complex 3, and here's complex 4. NADH actually is oxidized and donates its first pair of electrons in complex 1. It goes from this FMN to an iron sulfate protein, to a ubiquinone, to cytochrome B, to iron sulfate, cytochrome C1, cytochrome C, cytochrome A, cytochrome A3. This downward flow of electrons is extremely exergonic. The negative delta G is resulting at the end. Each transfer of electron is moving from one protein to the next and gains energy as it does. If FMN is oxidized or is reduced from gaining the electrons from NADH, then it becomes oxidized as it gives the electrons to iron sulfate. Iron sulfate becomes reduced when it gains those electrons. Then iron sulfate is oxidized and reduces ubiquinone. And once again, all the way down the protein chain until it reaches the oxygen. The oxygen is the final electron acceptor. This oxygen molecule gains those electrons and is completely reduced. It is now carrying those negative charges that allows it to combine with the hydrogen ions to create water. If you look at this complex of cytochrome proteins again, you will see that the cytochrome complex 2 is where FADH2 donates its electrons when it is oxidized. As it donates, it actually enters at a lower point on this electron transport chain. This is why only 2 ATPs are produced per FADH2 versus the 3 for NADH. I'll show you in a little while how that is a bit different also. As we watch this flash diagram, we can see the translocation of hydrogen ions across the membrane into the inner membrane space by the drop-off of the NADH coenzyme. The little spaceship guy, NADH, drops 2 electrons off at this cytochrome complex. As it drops the electrons off here at the cytochrome complex, they will be translocated down the complex. As they move down the complex, they will actually pump the hydrogen ions into that intermembrane space. As they pump those hydrogen ions into the intermembrane space, they in turn will start to increase the concentration inside that area. Okay, let's review this again. The whole purpose for cellular respiration to occur is so that we can stay alive. We need energy to stay alive. We have to have energy to stay alive. Our cellular energy is ATP. The way that we make ATP is by releasing energy from our food. So we consume our food to get the energy out of the food. The major primary force of this process is oxygen. And you know, we must have oxygen in order to stay alive. And so when we see this process happening, hopefully it'll all start to make a little bit of sense. We breathe oxygen. Oxygen is that electron acceptor. It's a major oxidizing agent. It causes electrons to be stripped from our food and carried to it. Well, all of those oxygens, all of those electrons stripped from our food are being carried by these coenzymes NADH and FADH2. These electrons are then donated to the electron transport chain. NADH becomes oxidized to NAD+. When it's oxidized to NAD+, NAD+ can now go back, pick up more electrons from more food sources to bring it to oxygen. And it's a never-ending cycle. As long as we are living, we need oxygen. We have to have oxygen in order for this process to occur. Remember, the citric acid cycle will not happen. We will not reduce NADHs if we do not have oxygen. Even though oxygen is not involved in the citric acid cycle at all, it is extremely important in oxidative phosphorylation. So we must have it in order to oxidize all these NADHs again. So here's what's happening. NADH drops off its electrons to this first protein complex. It is now oxidized, can go back and get more electrons from our food sources. Those electrons then will travel from protein complex 1 be carried through ubiquinone to complex 3, be carried through cytochrome C to complex 4, and then all the way down to the end of oxygen. Now, what about those hydrogen ions? Those hydrogen ions are actively pumped out into this intermembrane space. As those electrons travel down, we can see these hydrogen ions being pumped out, pushed into this intermembrane space. There are 3 complexes that it is pumped out for NADH. Complex 1, complex 3, and complex 4. This is occurring by a proton motive force, pushing those hydrogen ions around in order to, as long as those electrons are being carried down. What, honey? As those hydrogen ions are pumped across the membrane, they are doing this by the flow of the electrons through these protein complexes. Now if you notice, FADH2 enters at complex 2 a little bit lower, and so in turn it can only pump 2 hydrogen ions, or hydrogen ions across 2 pumps here instead of the 3 like NADH. This shows you how NADH actually produces 3 ATPs, whereas FADH2 only produces 2 ATPs, because that energy occurs from the flow of these hydrogen ions. As we are transporting electrons down the electron transport chain, these protons are pumping into that inner membrane space. As they accumulate in that inner membrane space, there are more and more of them out there, and we know what the concentration gradient's all about. It wants to push or come back from an area of a very high concentration to an area that is very low. But in order for those ions to come back across the membrane, they need a protein. The protein they come back across is called ATP synthase. This protein, notice the ASE here, is an enzyme. This ATP synthase, if you haven't figured it out, synthesizes ATP. It is the enzyme that allows ATP to be created. There is a binding site inside this ATP synthase enzyme for ADP and inorganic phosphate. The two bind to this enzyme. When the hydrogen ions come flowing back across this enzyme, it actually spins this enzyme like a turbine. And as it spins this enzyme, ATP is shot out on the other side. Now we start to see hydrogen ions coming into the matrix. Well, those hydrogen ions, being positively charged, are attracted to these oxygens that have received all of the electrons from the electron transport chain. And in turn, water is being produced. These two processes, electron transport chain and chemiosmosis, are coupled together. The electron transport chain is an exergonic reaction, all downhill. Chemiosmosis is an endergonic reaction, building ATP. The two of them, coupled together, allow us to make ATP via oxidative phosphorylation. Oxidative phosphorylation is the coupling of the electron transport chain and chemiosmosis together. This image here of this dam shows you how all of this water stuck behind this dam is contained. What happens when these gates open and the water rushes through allows for a tremendous amount of energy to be created. This is another image describing the same electron transport chain. But what this image allows you to see are all of these hydrogen ions accumulated in this intermembrane space. And this very high concentration of hydrogen ions out here and a very low concentration of hydrogen ions in the matrix allows for this electrochemical gradient to push these hydrogen ions from high to low. As it does so, it must come through this ATP synthase enzyme. As it travels across the enzyme, it spins the enzyme and allows for the production of ATP to occur. Here's another vision of it. What you can see here is this is the whole mitochondrion. Here's our outer membrane, the intermembrane space, the intermembrane, inner membrane, and then the matrix. In the matrix, we are going through the Krebs cycle. We are making all of this NADH and FADH2 by oxidizing our glucose. We've completely oxidized glucose and have reduced FADH2 and NADH. Both of those are in the matrix now, along with our oxygen molecules. These two coenzymes drop off their electrons to each electron transport chain. FADH at a lower level than NADH, but they are still dropping off their electrons, becoming oxidized, being carried back to the Krebs cycle to pick up some more passengers. As those electrons travel down this electron transport chain, the hydrogen ions build up in this outer membrane space. High concentration of hydrogen ions wants it to come back across the membrane through this ATP synthase enzyme, and in turn, as we watch this flash movie, what we are watching is the NADHs translocating those protons into the inner membrane space. As the electrons are carried down the cytochrome complex, you can see that they are pumping hydrogen ions into that inner membrane space before they combine with our oxygen. Watch these red electrons as they travel down. Each one of them translocates at this point, and this point, a hydrogen ion into this inner membrane space. As those hydrogen ions build up in the inner membrane space, we can see this concentration increasing drastically. As they increase drastically, then they're going to want to move back across into the mitochondrial matrix, and that is what stems ATP phosphorylation. So these reactions are coupled together. The electrons travel down the cytochrome complex and in turn pump out those hydrogen ions into the matrix. Now if it was an FADH2 dropping off their electrons, they would start here and only pump across this protein and this protein, and in turn only 2 ATPs would be made in the process. If we look at this ATP molecule in a little bit of detail, you can see or ATP synthase enzyme in a little bit of detail, you can see what I was talking about. And I actually have an animation that describes it as well. These hydrogen ions are building up in this intermembrane space. The concentration increases drastically, dropping the pH. What's going to happen are these hydrogen ions are going to come through this cylinder almost of this ATP synthase enzyme. This rod that it travels through will actually start to spin the overall complex of ATP, or of the ATP synthase. As those hydrogen ions flow down, it causes it to start to spin. As it spins, it actually causes this ATP to be manufactured or phosphorylated. If we return to the same flash movie complex here, you can see once again the outer membrane, the inner membrane with its folds of cristae. This is the intermembrane space. Now this intermembrane space is full of hydrogen ions. And what this video is now showing you is how those hydrogen ions flow back across the membrane through this enzyme known as ATP synthase. As these hydrogen ions flow across the membrane, it actually spins this ATP synthase and in turn produces ATP. If we look here, we could see here is an ADP molecule and an inorganic phosphate molecule. They enter into the enzyme. As the hydrogen comes across, it allows it to come back out in the form of ATP. Watch closely again as that happens. The ADP and inorganic phosphates both join into this enzyme together. The hydrogen ion spins it, and out comes ATP. Phosphate, ADP, out comes ATP. Phosphate, ADP, out comes ATP. If we look at the top angle of this ATP synthase enzyme, what you can see is how there are binding sites for ADP and inorganic phosphate. And as the spinning occurs, this is occurring because of the hydrogen ions coming through. When it spins, it allows the two to come together and out spits ATP. So watch, in comes ADP and inorganic phosphate, out comes ATP. So it's almost like the spinning of this rod allows this enzyme to activate the formation of ATP. So ADP and inorganic phosphate are being converted to ATP in this enzyme with the flow of those hydrogen ions. Let's look one last time at this flash movie process. Here we can see the whole dynamics happening at one time. The NADH comes in, drops off its electrons. Those electrons pump the hydrogen ions across the membrane, builds up the hydrogen ion concentration inside the membrane. As those hydrogen ions are building up, they want to come back across the membrane through the ATP synthase and in turn form ATP. This whole process is allowing the majority of all of our ATP to be produced. This is showing all of the coupled reactions of the electron transport system. NADH becomes oxidized as it loses those electrons to the first cytochrome complex. Those electrons are carried all the way to the end to oxygen. This yellow guy here is oxygen. Oxygen picks up those electrons, picks up the hydrogen ions in the matrix, and forms water. See how he has nothing? Picks up the electrons and hydrogens together. Those hydrogens come back across this ATP synthase enzyme and form ATP. As you watch this whole flash movie, you can see the whole process. It's a little confusing, but if you follow it enough, you can see the whole process going on over and over. And this is exactly what's happening in all of our mitochondria in our cells. This animation of the electron transport chain is a little less complex than what we were just watching, but this will also show the major events of the electron transport chain. For clarity, the protein complexes in the membrane are basically shown as blobs, and they are blue when they are reduced and red when they are oxidized. So that kind of helps let you follow along. Now as you look at this image, you could see that this cytosol is here where the glycolysis occurs. Here's the outer membrane, the intermembrane space, the intermembrane, and then the mitochondrial matrix inside. So as we go, we can take a deeper look at what's going on here. Here's those cytochrome complexes embedded within the membrane. This is where the electrons will be carried and the hydrogen ions will fill this intermembrane space out here. So let's look at this process. Here are the cytochrome complex proteins. Cytochrome 1, cytochrome 2, or 3, cytochrome 4. As the electrons are traveling down these cytochrome complexes, hydrogen ions will be pumped through proton motive force into the intermembrane space. So let's watch that process. NADH donates its 2 electrons and pumps hydrogen ions across that intermembrane space. When it does so, it is carrying those final electrons to oxygen. Remember, oxygen is the final electron acceptor. Let's watch that process one more time. Electrons donated all the way down to oxygen. Now recall, FADH actually enters at a lower point on the electron transport chain, and so it will only translocate 2 hydrogen ions. So as we drop off those electrons, 2 hydrogen ions will pump into that inner membrane space. Because of this, only 2 ATPs will be made through the ATP synthase enzyme. When those hydrogen ions are accumulated here in this inner membrane space, they want to come and travel down their concentration gradient and go through this ATP synthase enzyme. So let's watch as those protons travel back through the ATP synthase. Boom! Out comes ATP. Boom! Boom! So on and so on and so on. Remember, we have 10 NADHs and 2 FADH2s that are contributing to this process. And in turn, 34 ATP molecules will be made on the outcome. This high concentration of those hydrogen ions want to come back down the concentration gradient to the matrix where there are not as many hydrogen ions. When they come down that concentration gradient, it provides the energy for the ATP synthase to produce ADP. Now let's summarize cellular respiration. Cellular respiration begins with a glucose molecule in general. Glucose is a 6-carbon sugar molecule that through the first steps of cellular respiration is broken down into 2 3-carbon molecules known as pyruvate. Each of these are still organic molecules. The 2 pyruvates produced also give us a net gain of 2 ATPs and a reduction of 2 NADHs. Now these 2 pyruvates will enter into the citric acid cycle. As they go inside the mitochondrion, however, they have to go through a transition phase. This transition phase takes pyruvate and oxidizes off a carbon dioxide. In the end, we have 2 carbon dioxides produced and 2 acetyl-CoA enzymes. By oxidizing CO2, we also reduce 2 more NADH molecules. The Krebs cycle then takes this 2-carbon acetyl-CoA enzyme and oxidizes it completely to 2 more carbon dioxides. So for each pyruvate, you can see that 3 carbon dioxides are released. The Krebs cycle must occur 2 times because there are 2 pyruvate molecules entering into the mitochondrion. In turn, the citric acid cycle then gives us 4 carbon dioxides, 2 ATPs through substrate level phosphorylation, 6 NADHs, and 2 FADH2s. So glycolysis gives us 2 ATPs, which is great. The citric acid cycle gives us 2 ATPs, which is great. And if it all comes from just a simple investment of 2 ATPs, I've already made a 200% profit. However, most of the energy is still stored in the molecules of the NADH and FADH2. These coenzymes carrying those electrons still are storing a tremendous amount of energy. Because each NADH, through the process of oxidative phosphorylation, converts to 3 ATPs. And each FADH2, through the process of oxidative phosphorylation, gives us 2 ATPs. So if we look here, we see that we have 2 NADHs plus 2 NADHs, that's 4, plus 6 is 10 NADHs. So the 10 NADHs made through these first 2 steps of cellular respiration have the ability to be converted to 30 ATP molecules via oxidative phosphorylation. Each FADH2, since we have produced 2 of them, can be converted to 4 ATP molecules through oxidative phosphorylation. So when we look at this, we realize that 34 ATP molecules have yet to be made because they are stored in those NADH and FADH2 coenzymes. Once those electrons are carried to oxygen, 34 more ATPs will be produced, giving us a total of 38 ATP molecules in all. So if we look at it again, we can see that glycolysis gives us 2 ATPs via substrate level phosphorylation. But it will also give us anywhere from 4 to 6 ATPs for oxidative phosphorylation. Now the difference here comes with that NADH. In glycolysis, we're making or reducing NADH, but quite often when the NADH has to cross the membrane, it actually may be reduced to an FADH2 molecule, and in essence losing a little bit of its energy yield to produce only 2 ATPs per molecule instead of 3. The the transition phase or formation of acetyl-CoA gives us 0 ATP produced through substrate level phosphorylation, but it does produce 2 NADHs, which is a total of 6 ATP through oxidative phosphorylation. Because this acetyl coenzyme A is made in the mitochondrion, each NADH is already produced and it's not going to reduce to a lower form. The citric acid cycle also gives us 2 ATPs via substrate level phosphorylation. However, the 6 NADHs and 2 FADH2 gives us another total of 22 ATP molecules for a total of 24 ATPs phosphorylated via the Krebs cycle. Overall, that gives us a total of 36 to 38 ATP molecules in all. This is a tremendous amount of energy from one simple glucose molecule. Cellular respiration is about 40% efficient, which is a tremendous yield when you really think about it. So here to see it in a more schematic view or diagrammic view, we can see glycolysis breaks glucose down to 2 pyruvates, produces a net gain, remember that was our net gain of 2 ATPs through substrate level phosphorylation, and reduces 2 NADHs. The 2 pyruvates, if and only if oxygen is present, will enter into the matrix of the mitochondrion, going across the 2 membranes. As it goes across the 2 membranes, it produces acetyl-CoA, 2 of them, to enter into the citric acid cycle. This acetyl-CoA formation provides us with 2 more NADHs. The citric acid cycle produces 6 NADHs and 2 FADH2s, and 2 ATPs via substrate level phosphorylation. All of the NADHs and FADH2s are carried to the electron transport chain to go through oxidative phosphorylation. We see the variation once again up here with those 2 coenzymes coming in from glycolysis. They can either be NADH or at a lower energy yield of FADH2. In essence, we are making approximately 36 to 38 ATP molecules per glucose. Let's watch this entire process of cellular respiration. We start with a 6-carbon molecule known as glucose. Glucose goes through glycolysis and is split into 2 3-carbon molecules known as pyruvate. Through the process, those pyruvate, we see 2 NADHs being reduced, 2 ATPs total net gain being made through substrate level phosphorylation and 2 pyruvates. The 2 pyruvates then go through this transition phase, make 2 more NADHs, reduce to acetyl-CoA, and enter into the citric acid cycle. The citric acid cycle allows for 3 more NADHs to be reduced, 1 ATP to be phosphorylated, and 2 carbon dioxides are oxidized. We also get 1 FADH2. Now this process runs very quickly, so you need to pay close attention to what's going on throughout it all. I will let it run for just a few moments or a few cycles. Now sometimes oxygen is not present. When oxygen is lacking, either in our cells or in other organisms, we actually go through a process known as fermentation. Remember, oxygen is required for pyruvate to cross over the membrane into the mitochondrion to allow for the citric acid cycle to happen. If oxygen is not present, there is no sense of stripping pyruvate of all of its energy, and so cellular respiration does not proceed. Fermentation, on the other hand, will occur. Fermentation, alcoholic fermentation, is one type of fermentation. This is what yeast and some bacterial cells will go through, as well as a lot of our plant cells. When oxygen is lacking, the pyruvate is produced via glycolysis as normal. However, if there is no oxygen, the pyruvate will then be broken down and oxidize off a carbon dioxide. When it oxidizes the carbon dioxide, all that is left is acetaldehyde, that same 2-carbon group that we saw in the transition phase. However, there's no coenzyme A here. That acetaldehyde will then cause NADH to be reduced, or oxidized. So the hydrogens and electrons that were picked up during glycolysis will actually be put back onto the acetaldehyde, breaking this double bond here between this carbon and oxygen, and then attaching 1 hydrogen there and 1 hydrogen there, and in turn oxidizing NAD+. We now have produced ethanol, which is an alcohol. Needless to say, this is our alcohol that we drink in our beers and our wines and things like that. This carbon dioxide that is released is a gas that can actually be captured or maintained within those drinks. NAD+ is oxidized and can go back to glycolysis and be reduced again over and over and over. Now, animal cells do not go through alcoholic fermentation, or else we would probably all be exercising constantly with to get a lack of oxygen so that we had this automatic high from the alcohol. Not what happens, which is also a very good thing, because we actually go through what's known as lactic acid fermentation. Lactic acid is that burning sensation you feel within your muscle groups after a nice hard workout or a long walk, or even walking up to the top of the stairs sometimes and you start to feel that little burn. Because what's happening is there's not enough oxygen there to convert pyruvate to carbon dioxide. And so what happens is the pyruvate actually picks up the hydrogen ions and electrons from the NADH reduced during glycolysis, and the double bond on the number 2 carbon is actually broken, and we get the hydrogen and hydrogen placed on there. The good thing about lactic acid fermentation is that the NAD+ is oxidized, can very easily be reduced again bringing us back to pyruvate. And if we reduce it again back to pyruvate, once oxygen is present, that pyruvate can enter right back into the citric acid cycle. And so we are capable of bringing lactic acid back to pyruvate when oxygen becomes present. This pyruvate molecule then is the key junction in catabolism, in the breakdown of our food. If oxygen is present, we will continue to break pyruvate down. If oxygen is not present, we will be going into some form of fermentation, either ethanol or alcoholic fermentation, or lactate or lactic acid fermentation. Now, just to show you that carbohydrates are not the only source of energy for cellular respiration. Our carbs, our sugars, are the most direct route for cellular respiration to take place. As you can see, those carbohydrates go directly into glycolysis, producing pyruvate, acetyl-CoA, citric acid cycle, and oxidative phosphorylation. However, we can get energy from other food sources, like proteins and fats. If we look at fats, for example, remember our triglycerides contain one glycerol molecule and 3 fatty acids. If you've learned anything, I hope that you remember that those carbon and hydrogen bonds in those carbohydrate chains, or not carbohydrate chains, but hydrocarbon chains, are the key to cellular energy. The stripping of the electrons and hydrogen ions away from carbon. Our fatty acid chains are all hydrocarbon chains. They can be 15, 20, 25 carbons linked together. This is why our fats contain a tremendous amount of energy, 200 times more energy than carbohydrates. But our fats are our long-term storage units. We don't utilize them as long as carbohydrates are present. So those fatty acids, when we do utilize them, the glycerol easily enters into glycolysis as glyceraldehyde 3-phosphate. The hydrocarbon chains of our fatty acids then enter into the process at acetyl coenzyme A. If you recall, once we enter in here, we've actually lost 2 ATPs through the process of glycolysis and the 2 NADHs that were reduced. But that's okay because we're producing a tremendous amount of NADH per turn of the citric acid cycle. Imagine a fatty acid of 20 carbons in a row. That's 10 turns of the citric acid cycle, which gives us a tremendous amount of energy. But there is a flaw to this process. If we lived on a diet of fats alone, that oxaloacetate, which is required at the end of the citric acid cycle to begin the next citric acid cycle, can become depleted because it is not being produced. So it's kind of like what diabetics go through, which is, you can describe it easily as saying starving in a land of plenty. So you need to be careful when you're going on one of those all fat or all protein and fat diets, because you could start to show ketones in the urine and you could start to show some problems going on. Now we also can digest our proteins. Proteins are amino acids. They can enter into this chain of cellular respiration as at the pyruvate level, at the acetyl-CoA level, or directly into the citric acid process cycle. So we do lose the ammonia of our amino acid, the NH3, as a waste product, but we can generate some energy out of those proteins depending on the amino acid structure. Will depend on where it enters into cellular respiration.